Cell growth and division. So first question. We've talked a little bit about DNA this year. Not a lot. We know that cells come from other cells. Depending on which kind of cell you are, this can look a little different. But in the complicated cells that you are mostly familiar with, it's mitosis. In simple cells, like bacteria, right, prokaryotes, we call them, it looks different. It's much simpler. They just have a loop of DNA. They don't have a nucleus. So we just call it binary fission. They just kind of copy the loop of DNA and then split. Well, your division is more complicated than that. You have a whole mess of DNA to deal with. And you can't just copy it and then bloop, split in two, because it's like a whole big ball of yarn, and you're not going to get equal parts on both sides unless you organize things a little better. So technically, when we talk about mitosis, we're actually technically talking about the division of the nucleus, not the rest of the cell. It's the nucleus dividing that we consider to be mitosis. Two main jobs before division starts. Before you actually start, cutting it in two, two things you got to do with that DNA. First of all, you got to copy it. You got to copy the whole mess, the whole ball of yarn. Your cell goes through and copies all 6 feet of DNA. That's how much, by the way. You've got about 6. If you were to stretch it out end to end, 6 feet of DNA in each cell, in every single cell. And then once it's copied, then you have to organize it. Because like I said, it's when you're using it, it's like a big ball of yarn. Well, you can't just take a pair of scissors and cut that big ball of yarn that's two big copied sets of DNA and expect to get the right stuff on either side. You've got to organize it into these little X-shaped bundles called chromosomes. When it's in its loose ball form, we call it chromatin, and those X-shaped bundles are chromosomes. Every species has a distinct number of chromosomes. It doesn't mean a whole lot, but for what it's worth, your number is 46. You have 46 chromosomes, more like 23 pairs. And we'll talk about why we think of them as pairs in the next chapter. A chromosome, just to kind of give you the anatomy of one of these X-shaped bundles, we call the middle part the centromere. And this is the beginning of a whole bunch of C words here. Chromatin, chromosomes. Centromere is the middle part. If you divide that sucker right down the middle, the two halves you get are identical because these are doubled structures, right? We've replicated the DNA. We call those two halves sister chromatids. Questions so far? So where we'll eventually wind up here is what all the phases of this kind of division, this mitosis thing, what those phases look like. And you'll need to be able to recognize those phases and call them out by name and label the parts of the cells. But for now, we're going to talk about a larger chain of events, which includes mitosis, called the cell cycle. Every cell in your body, well, you tell me, is every cell in your body dividing? Why not?
Because they still have jobs to do.
Yeah, that would be chaos if all your cells were doing was just dividing all the time. If they all divided all the time, you'd just be a big blob of undifferentiated blob. What you are is a bunch of cells doing their jobs, making you, alive. And they're only dividing when they need to. Where would you expect to find cells in your body that are going through mitosis a lot? Good. Places where blood cells might be being made, like your Bone marrow is a place you can find a lot of cell division happening because your bone marrow produces blood cells. Your skin, even when you don't have a scrape, your skin has a layer of live cells, not on the outside. You know those are dead, right? That's your suit of armor. That's your waterproof seal and germ-proof seal, so stuff can't get in. It's cells that have died on purpose to protect you, but they're always coming off. You're sloughing off dead skin cells all the time. Did you know that like 80% of household dust is dead skin? you can't not know that now. But yeah, so you have to replace those dead cells all the time. So you got a layer of cells in your skin constantly going through mitosis. Absolutely. But then lots of other places, cells are just hanging out doing their jobs. So there's this cycle that all cells are in some stage of, called the cell cycle. And mitosis is just one little piece of that pie. Now, if you're something like a skin cell, you might constantly be spinning through this cycle. But a lot of other cells, most other cells, are hung up in some phase of this cycle, which I don't know if you should draw it until I get everything in it here. But everything in this cycle that's not mitosis is called interphase, which means the in-between phase, the not dividing phase. As soon as the cell is done dividing, its first problem is that it's half-sized, right? And so the first phase after mitosis is called G1 for growth. A couple of checkpoints in that phase, the cell has to get past, and then it starts getting ready to divide again by copying all its DNA. So it turns out that copying that DNA doesn't happen during mitosis, it happens in S phase. That S stands for synthesis of DNA, if that helps you remember. Then, sort of final prep for mitosis, making extra cell parts so the two new cells will have enough. Stuff like that. And then mitosis. And mitosis turns out to have 4 distinct phases. We call this M phase as a whole. And prophase, metaphase, anaphase, and telophase are the 4 phases of mitosis. And then there's an event that we kind of think of as the end of it, called cytokinesis. So that's the cell cycle. And it turns out there's actually a few places where a cell has to sort of pass checkpoints to go on. Otherwise, it's going to hang up in one of these phases. It's going to stick in one of these phases and just kind of do what its normal job is. These checkpoints are in 3 different spots. There's one at the end of G1, there's one at the end of G2, and there's one at the end of M. So you're going to want to label those as well. And you can do it, you can probably do a better job than I did. I had to come from a central point here. If you want to just label, checkpoint, 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 that's fine. But those are each 3 spots where a cell doesn't go any farther unless it has the right chemicals and the right equipment to do so. Part of this regulation system your body has to make sure you only produce new cells when you need them. How about one more example of where we might find dividing cells? And I'll give you a hint. It's going to be true of you and not of me. It's something that you're still doing. It's related to that. It's something that you're still doing that I'm not. Reagan?
Muscles.
Maybe. You're still growing. And that does include muscles. And you do find cell division, for example, when you exercise and you strain your muscles hard enough, and you work hard enough, your muscles do go through some mitosis and create new muscle cells. But yeah, like the ends of your bones, there's a lot of mitosis going on right now because you're growing. Whereas I'm done. Gravity's working on me and I'm probably getting shorter. Yeah.
Isn't there? Well, it depends on how your hair grows, but there's a lot of mitosis in your hair.
Absolutely. Your hair follicles are constantly dividing, and then those cells are dying as well.
cells as your hair and your nails. I feel like I've heard that before. Like, so,
Some of the proteins are definitely the same. Yeah, keratin for one is a protein that's in hair and nails. Yeah. Yep. Okay, so that's your kind of intro to cell division. We'll talk about a couple more things. I think Friday we'll talk why cells are so dang small, because there's an interesting mathematical reason for that. And then we'll get into these individual phases, and you'll get a look at cells in each of those phases and be able to recognize them. For now, I think we should color them. This is another page from that Best of All Coloring Books, the biology coloring book. And so the deal is the same. You don't just pick parts to color at random. You color each term a different color, and then you match the color of the term with the actual thing. And there's a little code in there, a little letter code that tells you which things are which. Okay, so I'm gonna get in your hand and then find my colored pencils. But you should have colored pencils, so get those out if you got them.